So I think we're going to get started. It's about that time for our next talk. Our next speaker is Dr. Rob Denton. So Rob Denton is an assistant professor at Marian University. He received his PhD in 2017 from Ohio State University and has spent the last decade studying salamander evolution and genomics, particularly of the all-female polyploid salamanders throughout the Midwest. And if you did not know, there are all-female salamanders. Kind of crazy, right? Rob's work with undergraduate students is supported by the National Science Foundation and the U.S. Fish and Wildlife Service. And I became first aware of Rob's work when I heard about tiny treadmills and salamanders, which I thought was crazy, but awesome at the same time. So thank you so much for joining us, Rob. I know I will learn a lot from you this afternoon.
Well, thank you, Ellen, and thanks to the rest of the Pierce Cedar Creek folks. I really appreciate the invitation, and I'll be delighted to come up there and see the salamanders on site here in a few weeks, hopefully, if spring ever comes there, which is in full spring, full swing here in central Indiana. I was asked to come and talk about some of the dispersal work that we've done with amphibians, but to back up just a little bit, most of this is focused on our interest in these weird animals you see here on the screen. So this might look like a blue-spotted salamander to you. It might look like a Jefferson salamander to you, depending on where you are. But this is a member of this unisexual, all-female group of mole salamanders in the genus Ambystoma. So no males, all female, and we define this by their single lineage of mitochondrial DNA. So that special piece of your genome that gets handed down from mother to daughter to daughter to daughter over generations. And at the same time, these animals have multiple copies of their nuclear chromosomes. So instead of us that might have one copy of chromosomes from mom and one from dad, these animals can have 3, 4, even 5 whole sets of chromosomes. And they've been around for a really long time. It's the oldest recognized vertebrate polyploid and all-female lineage. And one of the proposed reasons for why they've stuck around so long is they reproduce in a really unique way that, as far as we know, is only found in this group. Let me show you how this works from a salamander eye view. So this is one of these unisexual females. In this case, we're going to imagine that she is triploid. She has 3 sets of chromosomes shown here by these 3 circles. This female in the early spring, when vernal pools fill with water and we start to get some of these warmer, rainier nights, this female is going to come to a wetland and she's going to proceed to pick up a packet of sperm from a male of one of these other members of the genus Ambystoma. There's 5 possibilities here. I have 4 of them on the screen. She uses that sperm packet to initiate egg production. She lays those eggs, and most of them are clones of mom. The male contribution to these offspring oftentimes doesn't make it through. It's the same set of chromosomes that were in the mother. This isn't all that unusual. There are other all-female groups of vertebrates that reproduce this way, namely Amazon mollies in the southeastern United States and in New Mexico. What makes these animals different is the frequency at which that male contribution actually does sneak in to the next generation, either adding on an entire set of chromosomes or in some rare cases substituting for one that was there in the mother. And on paper, this is the perfect way to reproduce. These females don't have to make males of their own. They all get to directly have offspring, and they get to donate 100% of their genomic contribution to the next generation. It even has this very enigmatic name, kleptogenesis, so named after this stealing of sperm from males of other species. This is fascinating, and we ask lots of really interesting questions about these animals from genetic and evolutionary points of view. How do they manage this complicated genetic situation? These different sets of chromosomes, what are they actually doing for these animals in their cells? How does it affect their development? And it might not be immediately apparent what this has to do with conservation, but it turns out that these animals have several direct inroads to conservation of amphibians. And I'm going to talk about some of those specifically work that we did while I was in Ohio at Ohio State as part of the Ohio Biodiversity Conservation Partnership. I say this because they primarily steal sperm from males of these two species, Ambystoma lateral, the blue-spotted salamander, and Ambystoma jeffersonianum, the Jefferson salamander. I have their ranges here in purple and blue, and then the gray outline is the range of these unisexual polyploids. One of the direct conservation implications of this is the fact that where you might be looking at salamanders and thinking that they are one of these two sexual species, in fact, they might not be. So in the field, these animals are extremely difficult to distinguish from one another. And we have folks from all over the country send us samples every year to get us to do the genetic analyses to actually determine whether the animals that they are monitoring, helping cross the road, counting every year are actually the species that they care about, or is it this all-female lineage that might not have certain protections in their state? It might not be a species of concern. It might not be state threatened. These unisexual polyploids are absorbing this genetic information from these sexual species. So for instance, in places like Ohio, where Ambystoma lateral is a state threatened species, the most of the genetic information from animals that used to be in Ohio are captured in this unisexual lineage, not actually found in the few populations that we have left in the state. So in some ways, folks were interested in studying these as a proxy for the species that they're really concerned about, but also, what is the value of this? If you have this genetic legacy in this other group, does it matter, and how do we think about those issues? If you've seen some of this terminology, these letters is typically how we describe these different combinations of genomes that these unisexual polyploids might have. In this case, this animal is a triploid. It has 3 sets of chromosomes, 1 taken from Ambystoma lateral and 2 taken from Ambystoma jeffersonianum. And one of our most engaging questions that we've asked about these animals is a big one. How do they even coexist with one another? So if you're paying attention to talking about this reproductive system, there's a real conflict that's happening here. These females that make up this unisexual group require sperm from males of other species. At the same time, the males of these other species likely don't stand to benefit from their genetic information going into this unisexual lineage. What prevents them from just driving one another extinct? So these unisexuals can, on paper, reproduce at twice the rate because they have all females that are directly reproducing. Why don't they overrun the sexual species? And then why don't the sexual species just ignore them and not allow them to use their reproductive material? It's a really weird ecological and evolutionary question, but there is a really fantastic background of studies that ask this question: what types of life history characteristics would mediate these interactions? And so I'm going to show you 4 of these that come from the literature on sexual parasites, which this is sort of an example of. And I'm going to run down for each of these life history characteristics, which of these are an advantage for the sexual species, whether that's lateral, jeffersonianum, texanum, and which of these are advantages for the all-female group. And the first of which is mate choice. So you can imagine if the males of the sexual species are really good at telling the difference between this is a female of my own species and this is one of these unisexual salamanders, I should ignore them. This would be an advantage for that group, making these good reproductive behavioral decisions. And we have some evidence that that's the case. At least in Ambystoma texanum, these males can recognize their female of their own species versus the unisexuals and make that choice actively. So it's an advantage for the sexual species. Secondly is fecundity. And like many other all-female groups, these salamanders in this all-female lineage have much lower fecundity. So they lay fewer eggs, fewer of those eggs actually develop and successfully metamorphose out, and become adults. This is another advantage for the sexual species. In groups like amphibians that have this intermediate life stage in the water, that's an incredibly important time for setting them up for success when they leave the water. And so larval competition can be a potentially important part of these interactions between these competing species. And this is pretty much a wash. There's really no evidence in the one paper that's been published about this between this unisexual group and the sexual species. The fourth is dispersal. And this might seem like a weird one. What does dispersal have to do with this? But dispersal can be important for a couple of reasons. So one, if these unisexual salamanders are better at moving around and maybe taking advantage of naive males who aren't used to seeing them and can't make those good reproductive decisions, or they're better at moving around a landscape and utilizing more marginal habitat where they don't have to directly compete with the sexual species as much, this can allow them to maintain a foothold in these populations without having that kind of direct competition with the sexual groups. And unfortunately, we don't really know a whole lot about salamander dispersal. There's a decent body of work out there, but it's really hard to do. Very few of these animals disperse across the landscape. Most of them just return to the wetlands from which they came out, where they were born. The few that do disperse, how do you track an individual salamander over the course of years, relatively short movements? They only move for a short period of time when it's really nasty outside, is sleet and rain and cold. So this is a bit of a mystery, but is this something that's helping explain this situation? Because if we take the information on this slide and we plug it into some of these computational models that predict these interactions, 100% of the time it predicts that these unisexuals shouldn't be around. They should get outcompeted relatively easily, but we know that's not true. They're found all over the place. Oftentimes they're outnumbering these sexual species 2 to 1, 3 to 1 in some cases. They seem to be doing just fine and have been around for a long time. So we wanted to test this hypothesis. These unisexual salamanders have greater dispersal ability than a sympatric sexual species that's found in the same pond. So if we go out and we look, we're going to see that these unisexual salamanders move around either more frequently or farther than a sexual species that's sharing the same types of environments. So to do this, we went out to some sites in north central Ohio, in a very agricultural portion of Ohio in Crawford County. And specifically, the reason is that we knew a lot about the sites that were there. Within the certain area of that county, we knew where all the ponds were. We knew who owned those homes and those properties. We knew what species had bred there in the past, which ones were really vernal, which ones were permanent. And so we had a lot of information to be able to actually know where to look for all these. And nearly all of these areas in Crawford County that we were surveying looked something like this. So these beautiful vernal wetlands, usually in wooded lots behind people's farmhouses or barns. The reason why they weren't turned into farmland is because they habitually flooded every year and it became this wonderful breeding habitat for amphibians. But you could do a 180-degree spin and walk 10 feet, and suddenly you're looking at miles and miles and miles of agricultural land. So what you see here on the right, obviously a really intimidating landscape for a salamander that's trying to get around. We surveyed these sites to try to identify animals that have dispersed from the wetland they were born in. So we spent around 3 solid springs surveying 15 core wetlands in this area. And that ended up being genetic samples from about 150 of the unisexual salamanders and around 300 of the local sexual species. In this case, it's Ambystoma texanum. Then we took those tissue samples and we extracted DNA from them, and we used these species-specific pieces of information in that DNA to get some genetic information about them. So, for example, when we took a sample from one of these unisexual salamanders, we knew that the information we're looking at was from one of the specific sets of chromosomes that they had, and we could compare that to the corresponding information in texanum. And we used these approaches to try to see who was related to who in a relatively assumption-free way, and identify these individuals that like stuck out as sore thumbs. If we went to a site we genotyped all the animals that we captured. They're all closely related to one another because they're from the same pond. They were likely born there and then they came back to breed there. But every once in a while, you would find one that in fact isn't closest related to the animals we found it with in that pond, but was much more closely related to the animals that were from a population 5 miles down the road. At a certain statistical probability, we would call that animal a disperser, an individual that was assigned to a site other than where we sampled it with high confidence. And sure enough, we found some of these animals in the data that had a genetic signature of moving around the landscape. Not many of them. So a predictably low number here of the entire population. So you can imagine this makes sense because most of them return to the same wetland that they were born in. So we're going to have few to start with. And so we were lucky to even find some. And it's a similar number between these two groups. So nothing too shocking there. But if you look at the straight line distance from where we found the animal to where we actually assigned them, where they're from, that distance is much different between these groups. So it's about double for this sexual species, Ambystoma texanum, compared to the unisexual. So this suggests these texanum salamanders are moving much farther compared to the all-female group. To give you an idea of what that looks like in practice, I've put the sites here on a map. On the top are blue diamonds, which are the sexual species where we surveyed texanum. On the bottom are the orange diamonds, which are representing where we found the unisexuals. I'm going to map on here the movements between dispersed animals. So where we found them back to where we assigned them. And the important thing, and the arrows are going to be sized by the number of individuals that had the same path. Now, I want to be clear that this isn't the actual path that the animals walked, right? These are just arrows to demonstrate the magnitude of this difference. So on the top, dispersal routes from texanum are broadly really big. They're making these big movements all the way across this map from site to site, whereas the dispersed animals of representing this unisexual group are mostly just adjacent hops from one pond to the other that are next to one another. This information gets really silly if we try to predict the most likely route that they would have moved using something like landscape resistance models. This is such a harsh habitat to begin with all that agricultural land, that some of the routes they suggest are pretty wild and make these differences in terms of distance even much greater, to almost a comical degree. Now, this doesn't support this hypothesis. In fact, it's quite the opposite. There's a similar percentage of these dispersed individuals, and there's a greater dispersal distance for the sexual species, not the unisexual. So again, this is another advantage for the sexual group and doesn't necessarily answer the original question. But one idea that we thought about when we looked at this data was, well, why is this the case? Let's start thinking about physiology and why one group might be dispersing around much more than the other. And that's a complicated question. There's lots of things going on there. There's behavior, there's physiology, there's evolution, there's selection. And one of the simplest questions that an undergraduate in our lab actually came up with at a lab meeting was, well, what if they can just walk farther in a given amount of time? Very simplistic answer, but it bears a lot of explanation. These animals don't have a lot of time to move around the landscape, really only across these agricultural fields during heavy downpours in the evening during a certain time of year when temperatures are just right. Those animals that can move farther in that amount of time are just more likely to find these second breeding sites, a place where they can then breed and be dispersed to. So how do you test something like that? Locomotor endurance. Well, we were lucky to reach out to some colleagues that happen to have a salamander-sized treadmill that we could borrow to actually test locomotor endurance of these animals. So this is a small treadmill with the track of it is made out of wetsuit material, so you can keep it wet. We went out in the field, we grabbed adult animals from these same sites in Crawford County and brought them back to the lab and conducted these locomotor endurance trials, which will show you kind of what these look like here. Take an animal, put them on this treadmill, and you encourage them to walk on the treadmill, maybe poking their tail or their back foot. Eventually, they usually settle down into a consistent stride. They're not sprinters, right? And you, every 3 minutes, take an animal off the treadmill, flip it over on its back and see if it can right itself. If it can't right itself, you say that that animal is exhausted, it's lost its locomotor endurance. How long and how far did it travel before it got too tired to continue on? Those videos are really entertaining and people like to see them. But that work itself was actually really horrendous. And one of the reasons why is because these salamanders could walk on these treadmills for a really long time, particularly Ambystoma texanum, the smallmouth salamander. They walked around 4.5 times farther than the unisexual lineage. It's about 160 meters compared to around 130 meters. That might not seem like a big number, but if we scale the size of a salamander up to average adult human weight, that's like you or I jogging for 80 miles before we just get too tired to continue on. A pretty amazing feat for this chubby little animal with tiny stubby legs to walk that far. And this is what the data looks like. This is log distance on the y-axis. And then the two groups that we're working with here on the X, on the left is texanum, on the right are the unisexuals, and each of these dots is an individual's performance. The gray bar is the mean for these groups. So clearly a difference, but there's a couple interpretations of this. So one could be that the unisexuals are terrible at this activity and have very low locomotor endurance, or maybe the smallmouth salamanders are just particularly great endurance athletes. And we had a couple other species in the lab at the time to compare them to get a sense for how it compares to other members of Ambystoma. Not many to compare to, but it's clear that these values for Jeffersonianum and lateral are at least within the ballpark of Ambystoma texanum, supporting that these unisexuals actually had a depression in this ability compared to sexual species more broadly, not just the smallmouth salamanders. This is what I just showed you on the right, and then I'm going to compare it to the exact same type of figure made using the data from the genetic study. Each of these circles now is one of those disperser animals, and then on Y is the distance that they would have traveled. Same groups here on the X, and the gray bars are the means. And what makes this really fun science to do is because it's this example where you have a pattern in genetic data, the dispersed animals that we measure using only genetic information that matches a functional test, the locomotor endurance, that you would predict could produce that type of result in the genetic data. So these two things are likely relatively connected, and it's fun to make that connection from genetics to functional physiology. It didn't answer the question though, right? So if dispersal doesn't favor these unisexuals, well, what does? And we don't really know. And we're trying to continue to work on this over time. It's a fun question. One legacy of this work is that it is really suggested that there's a physiological consequence of this reproductive mode called kleptogenesis. For some things that we measure, like locomotor endurance, these unisexuals are terrible. They do not have the kind of locomotor endurance as the sexual species. They walk on the treadmill for 15 minutes and they're just too tired. They flip them on their back and they can't get up. For other things we measure, like tissue regeneration, for example, the unisexuals are superior to the sexual species. They can regenerate their tissues around 1.5 times faster than related sexual species that are diploids. What's going on here? What's the difference between these things? And it got us really thinking about the underlying genetic situation. For tasks that require whole body physiology, metabolic type things like producing energy when you're walking on a treadmill, this requires your mitochondria to be functioning well and requires some collaboration between your mitochondrial genome and your nuclear genome. Genes from each of those have to collaborate with the proteins that they make to be efficient. In contrast, things like generating new tissues is likely just a raw horsepower issue. You have more genes that produce more RNA that can produce more proteins and build these tissues. And so are we seeing this signal of conflict between mitochondrial and nuclear genomes in these salamanders? And the reason why that's such a compelling idea is that many of these unisexual populations are isolated and harboring these nuclear genomes that are far away from where they should be. So unisexuals there in southwestern Michigan have a Jeffersonianum genome. The nearest population of Jeffersonianum is at least central Indiana, if not more south than that. What is that genome doing? It's isolated there and likely has to interact with the mitochondrial genome to be efficient and keep these animals alive. Each of these unisexual salamanders is this weird combination of a really old and divergent mitochondrial genome and this rotating cast of new nuclear genomes that are being taken from whatever species they are their local with. What are the consequences of this, and how do they manage this scenario? This is something we're working on now. We've been fortunate to have NSF funding to work on this question, and this is a real active focus in my lab currently. So lots of folks to thank for this work from Ohio State and elsewhere, including all of my current folks here at Marian at the University of Minnesota Morris and funding. The one thing that I want to leave you with real quick is something a little bit different, just to show you and showcase how engaging these animals can be and how fun it is to talk about this kind of work with the public. When that regeneration study was published, we were really fortunate to have it picked up by some media outlets, including an article in the Washington Post. And this was the time period when they allowed anonymous comments on articles in the Washington Post. So we read the article with the student who was the lead author on the paper, and everyone was really delighted. And we thought, let's go down in the comments and see what the average person on the internet thinks of these really strange animals. A little bit terrifying, but sure enough, the very first comment, and I can't make this up, was someone had anonymously written us a poem about these salamanders. And I saved it and I go back to it all the time. And I love to show it to people whenever they ask me to talk about these animals. So I'm going to put it on the screen, let you read it, and then I'm happy to take questions mediated by Ellen afterwards. Thank you so much again for the invitation.
I love how you ended that with a poem. That was wonderful. I wanted to give people a few minutes just to read through it, or a few seconds. We do have a few questions that came in, and I think you kind of answered this, but I don't know if you entirely answered this. One question that came in was about the difference between sexual species and unisexual species and the dispersal differences. So they were asking, were these sexual species always moving these far distances, or do you think it might be in response to the unisexual species?
Ooh, that's a good question. I mean, it's likely that these sexual species have been interacting with the unisexuals for very long periods of time. It's not a new thing to have them on the same landscape. So I imagine that it's been a consistent idea for the genetic information, at least. That's likely capturing dispersal happening across many generations, right? So we could have animals that are 10 or 15 years old in that data set that dispersed a long time ago because we don't know how old they are, and we're just kind of comparing the signal and the genetic data. So it's likely not necessarily due to the unisexuals, because we think that other sexual species have a similar performance to those. But I'm not sure how that mechanism would work if it was happening. Still a good question.
Something to figure out maybe some other time.
Oh, yeah.
Another question that we had come through the chat, do you think that unisexual salamanders may be more or less susceptible to disease that might wipe them out?
Hey, that's a really good question, and it's something that we're working on right now. So there's evidence that in other clonal animals, that being clonal has its penalties, because you can't mix up your genetic information as freely as you do in a sexual species. And this is one of the advantages of sex. These animals sort of have a different prediction because they are getting this new genetic information from other species, where for an average a gene that's involved in immunity, for example, they would have more divergent copies of that gene than within a sexual species. And so this is something we're working on this year. We're doing swabs, BD swabs for everyone in the field when we're going out, and working with some folks in Vermont to try to get at that. I'm not exactly sure what the answer is, but the unisexuals, at least when they reach adulthood, are relatively hardy compared to the sexual species. It seems they really mostly just struggle in the intermediate stage. As an early embryo, especially if they can make it out of there, they're relatively competitive with the local sexual species.
This sounds like stay tuned, right?
Oh,
All right,
always. There's a whole career of questions to ask.
All right, so got another quick question from a self-proclaimed non-herpetologist. Could the unisexual species, or I'm never going to be able to pronounce the scientific name on this, but could they be considered a distinct species, and could they have protection under the Endangered Species Act?
Yeah, it's an excellent question. And in the past, there has been a discussion about whether it's worth considering giving them their own species name. But as you may be picking up on, they really defy a lot of the reasons we call something a species. So they're not quite hybrids. They're more distinct than what a hybrid would be, but they're more homogeneous than you would expect a species to be, because they do actively reproduce with these other groups. They're almost such a weird exception that it's not even, there's no category for them. And so we just typically call them a lineage. Now, that doesn't mean that they aren't given a name in some instances. So in Illinois, for example, they continue to call them by an older species name that was used to describe hybrids between Jeffersonianum and Lateral. And one of the reasons they do that is to give them protections. At least that's my understanding. And so in some ways, I think it's used as a more practical way to help provide that protection. But from a strictly like biological point of view, they're just so weird, we can't really put a name on them.
They defy being put in a box, right?
Certainly that.
Awesome. I'm going to give you one more question quick, and then we'll give people a break. Are there barriers to dispersal that may impede salamander movements? Are there some habitats they won't move through?
Yeah, and that might be I'm not necessarily an expert in that field, but there have been folks who thought very carefully about corridors and how to provide ways for animals to get this connectivity between wetlands, and especially in fragmented landscapes like the one I was talking about. Clearly, they avoid areas that are open, that are dry, and prefer things that are shaded, moist. But it's hard to get the kind of resolution to a salamander's movements, right? So that's a very small area, and the way that we classify those resistances tends to be a lot more coarse. And it's a hard thing to study. You can't really go out in the field and follow an individual salamander around and just see what it does. That would take you decades to get enough data to make conclusions. And so there are folks who are working really hard on those questions, and there's certainly differences, but it's also a very hard thing to implement and then measure. If you build a corridor, when will you see that effect? How do you go and measure it? How do you call it a success? Is a very hard applied conservation problem.
And one that we have to continue to strive for, right? Well, thank you so much, Rob. We really appreciate you sharing your knowledge and your videos and poetry.
Sure.
And we're gonna keep going on with our conference. So thank you for sharing with us.
Yep.
All right. So